In the fall of 2016, a 68-year-old woman named Patricia received a diagnosis that placed her in a clinical trial at a major oncology center. Non-small cell lung cancer, advanced stage. The trial involved a class of drugs called checkpoint inhibitors, specifically anti-PD-1 therapy. The treatment worked by removing a molecular brake on the immune system, theoretically allowing T cells to locate and destroy cancer cells that had been hiding from them. There were 41 patients in Patricia's cohort, same drug, same dosing, same cancer type, but their outcomes diverged dramatically over the following months. About a third of the patients had significant tumor regression. The others showed little to no response. Patricia was somewhere in the middle, waiting, watching, not knowing which group she would end up in. Before the trial began, every patient had been asked to submit a stool sample. The researchers stored them without telling patients why. They thought the samples might be useful later. They were right, in a way that no one fully anticipated. Before we continue, a necessary pause. This is educational information based on peer-reviewed research published in Science and Nature Medicine. It is not medical advice, and it is not a treatment recommendation. If you have cancer or are currently in treatment, the conversation I'm about to walk you through belongs with your oncologist before it becomes anything you act on. I'm Dr. Becker, health researcher and science translator. My job is to read the literature so you don't have to, then tell you what it actually says. The stool samples told a story the researchers hadn't expected. When they analyzed the gut bacteria from every patient using a technique called 16S rRNA sequencing, which identifies bacterial species from their genetic material without needing to culture them, they found something that predicted immunotherapy response more clearly than tumor mutation burden, patient age, or cancer staging. One bacterial species showed up consistently and abundantly in the patients who responded to treatment. It was nearly absent in the patients who didn't respond at all. The bacterium is called Akkermansia muciniphylla, and what it does inside the human body, how it connects to cancer immune surveillance, to a specific fatal vulnerability in cancer cell metabolism, and to the question of whether your immune system can recognize a tumor, is what the next 18 minutes are about. Patricia's stool sample. The researchers flagged it early in their analysis. We'll come back to what they found. Let me start with what changed the scientific conversation about gut bacteria and cancer outcomes. In 2018, two independent research teams published back-to-back papers in the same issue of Science, the most prestigious peer-reviewed journal in science, arriving at the same conclusion from two different cancer types and two different institutions. The first paper was by Rowdy and colleagues at the Gustave Roussy Cancer Center in France and the University of Toronto. They studied 249 patients with non-small cell lung cancer, renal cell carcinoma, and urothelial carcinoma, all receiving anti-PD-1 checkpoint inhibitor therapy. What they found was this: patients whose gut microbiome was enriched with Akkermansia muciniphylla had significantly better progression-free survival and overall survival on immunotherapy compared to patients with low or undetectable Akkermansia. The bacterium wasn't a minor variable. It was one of the strongest single predictors of treatment response in their entire dataset. Honestly, when I first read this paper, I went back and checked the methodology carefully. The connection seemed almost too clean. Bacterial abundance predicting cancer drug efficacy? I wanted to know how robust the finding was. The answer: they replicated it. They took stool from Akkermansia-rich responders and Akkermansia-depleted non-responders and transplanted it into germ-free mice with transplanted human tumors. The mice receiving microbiota from human responders had better tumor control when treated with anti-PD-1 therapy. The mice receiving microbiota from non-responders did not. Causality demonstrated in controlled conditions. The second paper, by Gopalakrishnan and colleagues at MD Anderson Cancer Center, studied melanoma patients receiving anti-PD-1 therapy. Same pattern from a different direction. Responders had significantly higher gut microbiome diversity. They were specifically enriched with Faecalibacterium prausnitzii and related butyrate-producing bacteria. Non-responders had what the researchers called a dysbiotic microbiome, low diversity, low Akkermansia, dominated by bacteroidal species. And again, germ-free mouse experiments confirmed the causal relationship between microbiome composition and immune response to the drug. Two research groups, two cancer types, two institutions, same journal issue, same fundamental conclusion. Your gut bacteria are not passengers. They are co-regulators of your immune system's ability to fight cancer. Now let me explain what Akkermansia muciniphylla is actually doing, because the mechanism is more specific than the phrase good bacteria suggests, and the specificity is what makes it matter. Akkermansia muciniphylla is a gram-negative anaerobic bacterium that lives specifically in the mucous layer of the colon. It is one of the most intensively studied bacteria in the human microbiome. In healthy adults in their 40s and 50s, it typically makes up 1 to 3% of gut bacterial abundance. After age 60, its relative abundance tends to decline, progressively, along with gut barrier integrity, immune regulation, and metabolic function. This decline isn't random. It maps directly onto the environmental changes most common in the 55 to 75 age range. Repeated antibiotic exposure, chronic NSAID use, reduced dietary fiber, and altered gut pH from proton pump inhibitors. The loss is systematic and cumulative. What Akkermansia does in the gut, it metabolizes mucin, the protein component of the mucus layer. This sounds like it would thin the barrier, eating the wall. But the opposite is true. Akkermansia's activity at the mucus layer stimulates the intestinal epithelial cells to continuously produce fresh mucus and to tighten the tight junctions between cells. Think of it as a contractor who renovates the building while living in it. The structural activity triggers a constant repair response, so the barrier ends up stronger, not weaker, than it would be without the bacterium present. The result is a more intact gut barrier. A gut environment where bacterial fragments, endotoxins like lipopolysaccharide, LPS, stay contained within the colon rather than crossing into systemic circulation and triggering the chronic, low-grade inflammatory state that characterizes so many aging-related diseases, including cancer progression. The immune training effect is the second piece, and this is the one I find most underappreciated in the popular conversation about gut health. The gut-associated lymphoid tissue, the immune infrastructure of the gut, is in continuous communication with the systemic immune system. It's not a one-way broadcast. It's a constant bidirectional dialogue that shapes the entire immune landscape of the body. A gut environment with Akkermansia present maintains a specific distribution of immune cells at this interface. More regulatory T cells, more trained dendritic cells, better calibration between tolerance and attack. This calibration is not abstract. It is the specific immune architecture that checkpoint inhibitor therapy depends on to function. When Akkermansia is absent and the gut barrier becomes permeable, the systemic immune system shifts toward chronic, non-specific activation, less precision, less ability to mount the targeted T-cell response that anti-PD-1 therapy requires. This is part of why gut microbiome composition predicts immunotherapy response so consistently. The gut isn't adjacent to the immune system, it trains it, every day, based on what's living in it. In the Rowdy 2018 paper, the specific mechanism they identified was IL-12 secretion, interleukin-12, a signaling molecule that acts like a deployment order for one specific class of immune cells. Akkermansia-stimulated dendritic cells produce more IL-12, which in turn recruited CCR9-positive CD4+ T cells into the tumor microenvironment. These are the T cells that anti-PD-1 therapy is designed to unleash. The drug removes the molecular brake, but without Akkermansia priming the dendritic cell activation step upstream, there's no workforce to deploy when the brake is lifted. The drug signals, nothing responds. That is the precise failure mode in non-responding patients, and the Rowdy paper documented it at the cellular level. This is the mechanism Patricia's stool sample was pointing toward. Her Akkermansia levels placed her in the group where that upstream priming was compromised. We'll come back to what she did next. The second mechanism is entirely separate from the immune axis, and it involves a vulnerability in cancer cell metabolism that has been in the peer-reviewed literature since 2012, but is rarely discussed outside of academic circles. I find the silence around this one genuinely puzzling, because the logic is almost elegantly simple once you understand it. Certain bacteria in the gut produce a short-chain fatty acid called butyrate. The primary producers are Faecalibacterium prausnitzii, Roseburia intestinalis, and Clostridium butyricum. These bacteria ferment dietary fiber, specifically resistant starches and non-digestible polysaccharides, and release butyrate as a metabolic byproduct into the colon. The production is not passive. It requires the right bacteria, the right substrate, and a gut environment intact enough to support those bacterial communities. All three of those conditions are under pressure in the aging gut. Normal colon cells use butyrate as their primary fuel. They absorb it and oxidize it through the mitochondrial pathway, the clean, efficient energy production route, burning through it before it has any opportunity to accumulate. The colon wall is essentially a butyrate-consuming machine. The system works the way it was designed to work, and normal cells are well protected from butyrate at any concentration the gut naturally produces. Cancer cells are a different story. Cancer cells have altered their energy metabolism in a way that has become known as the Warburg effect, after the German physiologist Otto Warburg, who described it in the 1920s. Cancer cells preferentially generate ATP through glycolysis, the fermentation-like pathway, even when oxygen is abundantly available. They do this not because glycolysis is more efficient, but because it produces metabolic intermediates that support rapid cell division, biosynthetic building blocks for nucleotides, lipids, and amino acids. The trade-off is energy efficiency. Cancer cells are less efficient at producing ATP, but they prioritize speed and biosynthesis over efficiency. This metabolic shift is one of the hallmarks of malignancy and has been recognized for nearly a century. Here is where the vulnerability appears, and this is the part I find most remarkable about this entire research area. Because cancer cells have shifted to glycolysis, they cannot efficiently absorb and oxidize butyrate the way normal colonocytes do. Instead of being metabolized for energy before it can accumulate, butyrate sits inside the cancer cell. It enters the nucleus. And what butyrate does in the cancer cell nucleus is something cancer cells fundamentally cannot tolerate. Butyrate is a histone deacetylase inhibitor, an HDAC inhibitor. To understand why that matters, histone deacetylases are enzymes that compact chromatin by removing acetyl groups from histone proteins, silencing the genes packaged within. Cancer cells depend on HDAC activity to silence tumor suppressor genes, to silence pro-apoptotic genes, the genes that would tell the cell to self-destruct, and to silence the DNA damage response genes that would otherwise trigger the cancer cell to stop dividing and die. When HDAC activity is blocked by accumulated butyrate, those silenced genes become accessible again. The cancer cell's own suicide programs reactivate. The cell cycle halts. Apoptosis begins. What Donohoe and colleagues documented precisely in a 2012 paper in Cell Metabolism was this. The Warburg effect, usually described as a metabolic growth advantage for cancer, is simultaneously the mechanism that makes cancer cells selectively vulnerable to butyrate-induced apoptosis. The metabolic shift that makes cancer cells grow faster is the exact same shift that prevents them from detoxifying butyrate before it reaches their nucleus. Normal cells consume butyrate before it can act as an HDAC inhibitor. Cancer cells accumulate it, and that accumulation triggers their own silence destruction programs to come back online. Put plainly, the thing that makes cancer cells dangerous is the exact same thing that makes them vulnerable. And the vulnerability requires butyrate, which requires specific gut bacteria, which require dietary fiber and a gut environment that supports those bacterial communities. Let me be concrete about what that looks like in practice. A colorectal cancer cell sitting in a high butyrate environment, produced by a gut with adequate Faecalibacterium prausnitzii and enough dietary fiber to fuel it, is absorbing a molecule that its own metabolic architecture cannot safely handle. The HDAC inhibition that follows is not a side effect. It is a direct consequence of the cancer cell's identity. It chose glycolysis. It cannot unchoose it when butyrate arrives. That metabolic commitment, which gave the cancer cell its proliferative edge, becomes the mechanism of its own undoing. The research community has known this since 2012, and it has not yet made its way into the standard clinical conversation about colon cancer prevention. That gap is part of why I cover this. Raymond, 71, had been following his gastroenterologist's advice to increase dietary fiber for 2 years before his colorectal polyp surveillance scope. His previous scope at 69 had identified 3 adenomatous polyps, the precancerous type. At 71, the follow-up scope found none. His gastroenterologist, reviewing Raymond's dietary history and the research he had printed out from the published literature, noted that his fiber intake and targeted probiotic use would be expected to significantly increase colonic butyrate production. One case, one data point. The direction is mechanistically consistent with what the 2012 Cell Metabolism paper described. Raymond's gastroenterologist didn't make causal claims, but she noted the direction and kept it in his file. The practical implication runs directly from the mechanism. If butyrate selectively drives cancer cell apoptosis, and if butyrate production depends on specific gut bacteria that ferment fiber, and if those bacteria require a gut barrier environment that Akkermansia helps maintain, then the 2 things that matter most are feeding the butyrate-producing bacteria consistently and maintaining the Akkermansia-supported gut environment that allows those bacterial communities to thrive. One bacterium enabling another class of bacteria, a system within a system. In 2022, De Rosa and colleagues published a dedicated study in Nature Medicine, specifically examining Akkermansia muciniphila as a predictor of response to pembrolizumab, one of the most widely used PD-1 checkpoint inhibitors in oncology, in 338 patients with advanced non-small cell lung cancer. The finding was precise and independent. Patients with detectable Akkermansia muciniphila before starting immunotherapy had significantly better progression-free survival and overall survival than patients without it. After adjusting for age, gender, tumor mutation burden, PD-L1 expression, the protein that anti-PD-1 drugs target, and other clinical variables, Akkermansia abundance remained an independent predictor. It was not confounded by any of the other variables they controlled for. It was a standalone predictor of whether the drug would work. That distinction matters enormously. It means the bacteria are doing something that no other measured clinical variable is capturing. The same paper documented the antibiotic effect with a clarity that, when you think through the implication, is difficult to sit comfortably with. Patients who had received broad-spectrum antibiotics in the 2 months before starting immunotherapy had dramatically worse outcomes on pembrolizumab. Not marginally worse, dramatically. The mechanism is not complicated. Antibiotics don't selectively eliminate pathogenic bacteria. They eliminate commensal species simultaneously, including Akkermansia, including Faecalibacterium prausnitzii, including Roseburia intestinalis, including the entire butyrate-producing community that supports immune function and gut barrier integrity. The microbiome does not recover quickly after a course of broad-spectrum antibiotics. Full restoration of disrupted species takes months, sometimes considerably longer, depending on diet and the degree of initial disruption. Patients entering immunotherapy with a recently antibiotic-depleted gut were starting the treatment with a fundamental structural deficit in one of the key mechanisms that makes the therapy work. This has implications that extend well beyond active cancer treatment, and I want to be direct about it. Most people in the 55 to 75 age range have received multiple courses of broad-spectrum antibiotics over the preceding decade, for respiratory infections, for dental procedures, for UTIs. Each course causes partial microbiome recovery, but not full restoration. The species most difficult to re-establish after antibiotic disruption are often the exact ones most relevant to immune function: Akkermansia, Faecalibacterium, Roseburia. The cumulative impact is real and documented. The microbiome of a 70-year-old who has had 5 or 6 antibiotic courses in the past decade is measurably different from the microbiome of a 40-year-old with the same genetic baseline. It's not a minor difference. It's a structural difference in the immune training environment of the gut. What else depletes Akkermansia specifically? The list, when you put it together, tells a story about the standard medical management of aging in Western countries. NSAIDs, ibuprofen, naproxen, aspirin at high doses, alter the gut mucus layer and are consistently associated with reduced Akkermansia abundance across multiple population studies. Proton pump inhibitors, omeprazole, pantoprazole, and their entire class, alter gut pH in ways that create an environment less hospitable to acid-sensitive anaerobes, and several microbiome analysis studies have shown reduced Akkermansia counts in PPI users. Ultra-processed food dominance depletes the fiber and mucin substrate that Akkermansia depends on for fuel, while simultaneously feeding the inflammatory competing species that displace it. And chronic stress, through its effects on gut motility, mucus production, and the composition of gut secretions, has been associated with Akkermansia depletion in animal models, though the human data here is still emerging. The pattern is uncomfortable and specific. The standard pharmaceutical interventions, most common in the 55 to 75 age group, NSAIDs for joint pain, PPIs for acid reflux, antibiotics for infections, are collectively eroding the bacterial species most predictive of cancer immune surveillance capacity. Whether or not someone is in active cancer treatment, this is the background condition of the gut microbiome for a large proportion of the senior population. Honestly, this is the part of the research that I've thought about most since reading through it. Because none of those interventions are unreasonable in isolation. NSAIDs manage real pain. PPIs manage real reflux. Antibiotics treat real infections. But the aggregate effect on the immune training environment of the gut has not been part of the conversation, and the research now makes a strong case that it needs to be. Now let me tell you what happened with Patricia's stool sample. When the researchers in her trial ran the microbiome analysis, they found that Patricia's gut Akkermansia levels were low, below the threshold that had been associated with immunotherapy response in the French cohort. The analysis also showed reduced Faecalibacterium prausnitzii, the primary butyrate producer. Both of the mechanisms I've described were compromised simultaneously. She had been on a PPI for 18 months for acid reflux. She had completed a course of broad-spectrum antibiotics 4 months before the trial began, for a dental procedure. Standard care. Reasonable interventions at the time. But the cumulative effect on her gut microbiome had left her starting immunotherapy in a depleted state. The analysis explained the pattern. It also gave her a target. Patricia, on her own and with her oncologist's knowledge, started reading the 2018 Science papers and then the 2022 Nature Medicine study. She brought them to her oncologist and her gastroenterologist. She added specific dietary interventions to her routine: daily resistant starch from cold-cooked and reheated potatoes. The reheating cycle increases resistant starch content by restructuring the starch molecules. Daily leeks and garlic as inulin sources. Weekly consumption of pomegranate and dark berries for polyphenol content. And after discussion with her gastroenterologist, a pasteurized Akkermansia supplement. Her oncologist tracked her gut microbiome composition via stool testing at 3-month intervals. They were doing something rare in standard oncology practice, measuring the variable they were trying to move in real time. At 9 months from trial enrollment, Patricia's tumor showed a partial response, a reduction in volume that her oncologist called clinically meaningful. Her Akkermansia levels had measurably increased from the depleted baseline. Her Faecalibacterium prausnitzii had also recovered toward normal range. Whether the microbiome intervention contributed causally to the partial response is something no single case can establish. What the published literature can establish is that the mechanism is documented, the studies are consistent, the population-level data across hundreds of patients points in the same direction, and the intervention is low cost and low risk when implemented with physician awareness. Here is what the evidence specifically supports for rebuilding Akkermansia muciniphilia and maintaining the butyrate-producing bacterial community. And I want to be as specific as the research allows, because specificity is the difference between a protocol that works and a vague gesture toward eating better. First, prebiotic fiber, the non-digestible carbohydrates that feed both Akkermansia and butyrate-producing species. Resistant starch is the most important category here. It comes from cold-cooked potatoes, cooked and cooled rice, green bananas, and legumes, particularly lentils and white beans. The cooling step is not optional. When starch gelatinizes during cooking and then retrograded during cooling, it becomes structurally resistant to digestion in the small intestine and reaches the colon intact, where Akkermansia and Faecalibacterium ferment it. One cold cooked potato, eaten daily as part of a meal, provides meaningful resistant starch at a dose consistent with the dietary patterns associated with higher Akkermansia abundance in observational studies. Beyond resistant starch, inulin and fructooligosaccharides from Jerusalem artichoke, chicory root, garlic, onion, and leeks. These are particularly well studied as Akkermansia substrates. 1 to 2 cloves of raw or lightly cooked garlic daily, combined with a quarter cup of cooked leeks, represents a practical daily dose that doesn't require supplements. Arabinoxylan from oats, whole wheat bran, and psyllium husk rounds out the fiber profile. Psyllium husk in particular has been studied for its effects on gut barrier integrity and shows complementary effects with Akkermansia on mucous layer thickness. 1 teaspoon of psyllium husk mixed into water or yogurt in the morning is a low-friction way to add this consistently. The cumulative fiber target that the research associates with improved microbiome diversity in the 55-plus population is 30 to 35 grams per day. Most people in this age group are getting 12 to 15. The gap is not addressed by adding one food. It requires a systematic shift in what fills most of the plate at most meals, toward legumes, vegetables, whole grains, and nuts, and away from ultra-processed foods that provide no fermentable substrate whatsoever. Second, polyphenols, the plant compounds most consistently associated with Akkermansia growth in controlled intervention studies, and the category most people underestimate. Cranberry polyphenols have been shown in multiple randomized studies to increase Akkermansia abundance specifically. A 2022 study in MPJ Biofilms and Microbiomes showed that cranberry extract supplementation increased Akkermansia abundance in overweight adults, independent of dietary fiber intake, suggesting the polyphenol effect is not merely a proxy for fiber. Pomegranate extract and the urolithins produced when gut bacteria metabolize pomegranate elagitanins also stimulate Akkermansia, and this is one case where the metabolite is doing the work, not the parent compound. Resveratrol from dark berries and grape skin shows positive associations with Akkermansia in microbiome studies, as does EGCG from green tea, which has been studied specifically for its mucus-stimulating effects on intestinal epithelial cells. The practical application here is not a supplement stack. It is a consistent daily intake of deeply colored fruits and vegetables, a quarter cup of blueberries, a small handful of pomegranate seeds, 2 cups of green tea, as a regular part of the dietary pattern. The polyphenol effect appears to be cumulative and consistent, not spike-dependent. Third, pasteurized Akkermansia supplementation. A 2017 paper in Nature Medicine by Plovier and colleagues established something that changed the supplementation conversation significantly. Pasteurized Akkermansia muciniphila, bacteria killed by gentle heat treatment, retains full biological activity. The cell wall protein that drives the gut barrier and immune effects, called AMAC_1100, survives pasteurization intact. Pasteurization makes the bacteria shelf-stable and eliminates the viability problem that kills most live probiotics in stomach acid before they reach the colon. Several commercial products now offer pasteurized Akkermansia specifically, and this is the format with the most direct clinical evidence for gut barrier restoration. A 2019 proof-of-concept study in Nature Medicine by Despommier and colleagues, the first human trial of pasteurized Akkermansia, showed that 3 months of supplementation in metabolically unhealthy adults improved insulin sensitivity, reduced liver inflammation markers, and increased gut Akkermansia abundance, with an excellent safety profile. If you are considering Akkermansia supplementation, specifically in the context of cancer treatment or immunotherapy, this is a conversation that requires your oncologist's involvement, not because the supplement is dangerous, but because the research is moving fast enough that your oncologist may have updated protocols, may want to track your microbiome alongside treatment, or may be aware of contraindications specific to your treatment regimen that I cannot account for here. Fourth, be strategic about antibiotic use. Not all antibiotic prescriptions are avoidable, And I want to be clear that I am not suggesting you decline antibiotics for serious infections. But for routine infections where narrow-spectrum alternatives exist and where watchful waiting is clinically reasonable, the difference in microbiome impact between antibiotic classes is significant. A 5-day course of amoxicillin has considerably less impact on Akkermansia and Faecalibacterium than a 5-day course of a broad-spectrum fluoroquinolone like ciprofloxacin. This is a conversation worth having with your prescribing physician if you are actively trying to maintain or rebuild your gut microbiome. Not a confrontation, a conversation. Is there a narrower spectrum option for this particular infection? Is a reasonable question for any patient to ask. And if you do require a broad spectrum course, because sometimes there is no alternative, the research suggests that resuming high-dose prebiotic fiber and polyphenol intake immediately after completing the antibiotic course, rather than waiting weeks, shortens the recovery window for commensal species. Akkermansia is particularly sensitive to the post-antibiotic environment. If the gut is reseeded quickly with fermentable substrate and polyphenol stimulation, the species recovers faster than in a fiber-depleted environment. The bacteria need raw material to work with. A gut cleared of its microbial community and then starved of fermentable fiber is a gut that takes considerably longer to rebuild than one that is aggressively refed from day one post-antibiotic. The connections between these four strategies are not coincidental. Dietary fiber feeds Akkermansia directly. Polyphenols stimulate Akkermansia growth through a separate pathway. Pasteurized supplementation directly restores the species when diet alone is insufficient after significant disruption. And antibiotic stewardship protects the restoration once it has been achieved. Each strategy reinforces the others. The fiber without the polyphenols is less effective. The supplementation without the dietary substrate to support the bacteria post-colonization is less durable. The combination works because the gut is a system, and the interventions address the system at multiple points simultaneously. What to avoid? Beyond antibiotics, the specific behaviors most consistently associated with Akkermansia depletion in the research include ultra-processed food consumption at any significant frequency, chronic NSAID use beyond what is medically necessary, and prolonged PPI use without periodic reassessment of whether the original indication still requires medication-level management. For patients on PPIs for acid reflux, there are dietary and behavioral strategies: meal timing, head elevation during sleep, reducing late-night eating, that can reduce dependence on medication-level acid suppression. This is a conversation with your gastroenterologist, not a unilateral decision, but it is a conversation worth initiating. The timeline for rebuilding Akkermansia is not a 2-week protocol. The Depommier 2019 trial used 3 months of supplementation before seeing measurable changes in gut barrier markers. Meaningful species restoration after significant microbiome disruption, several antibiotic courses, years of PPI use, decades of low-fiber eating, takes 3 to 6 months of sustained dietary change minimum. Patricia's first measurable Akkermansia recovery appeared at her 3-month stool test. Her oncologist's assessment of meaningful tumor response came at 9 months. This is not a supplement you take for a month and evaluate. It is a shift in what you feed your gut consistently, long enough for the bacterial community to re-establish the critical population density required to train immune cells and maintain the gut barrier. Consistency across months is the variable that separates people who see results in the research from people who don't. If you are currently in active cancer treatment or considering immunotherapy, bring this information to your oncologist before making any microbiome changes. Some oncology centers, particularly the major academic centers where this research originated, are now actively tracking gut microbiome composition as part of immunotherapy protocols. This is a field moving quickly enough that your oncologist may have updated recommendations that post-date everything I've described today. The connection between the gut and cancer is not a metaphor, and it is not emerging science at this point. It is documented mechanism, replicated across institutions, confirmed in controlled animal models, and now tracked in prospective human trials. The bacteria in your colon are training your immune system's ability to recognize and respond to cancer cells. They are determining whether the short-chain fatty acids reaching your colon wall are accumulating inside cancer cells and triggering their apoptosis. In the context of immunotherapy, they are predicting whether the drug your oncologist is prescribing will find a competent, primed immune system waiting to receive the signal it sends. Akkermansia muciniphylla is not the entire story, but it is the bacterium the research keeps returning to. In Gustave Roussy, in MD Anderson, in Nature Medicine, in Cell Metabolism, across cancer types, across treatment classes, across institutions that had no reason to agree and did anyway. And rebuilding it starts with what you eat. Cold cooked potatoes, leeks and garlic, pomegranate, green tea, consistent daily fiber, and where appropriate, after discussion with your physicians, pasteurized supplementation to accelerate what diet alone takes longer to accomplish. Patricia's partial response at 9 months was one data point. The literature behind it represents thousands of patients and 15 years of converging research. The direction is consistent, the mechanism is established, the intervention is accessible. That is the background research, that is the mechanism, and that is the case for treating gut microbiome composition, not as a wellness trend, but as a functional variable in cancer immune surveillance that deserves the same serious attention your oncologist gives to tumor mutation burden and PD-L1 expression. Keep investigating, Dr. Becker.